Okay, so let's go ahead and wrap up the cell cycle and the cellular reproduction lecture. This is the fourth part. This will be just kind of a short one, just to go over some just review the basics, but also point out how bacterial cells, single-celled organisms, use this cycle or this type of cycle for reproduction of themselves. So remember, we've talked about bacteria a little bit when we were discussing cell structure and the cells. They are prokaryotic organisms. They do not have a nucleus. They have a nucleoid region. They're never going to be multicellular, single-celled. So in order for them to reproduce, they use a process very similar to our cell cycle, or our cell cycle is very similar to their reproduction, whichever way we want to look at it. Their process is called binary fission. Okay, so this is how bacterial cells make copies of themselves through this binary fission. Alright, so again, single-celled organisms, they do it a little bit simpler, easier, but it's completely effective. So what you're looking at here is actually a picture of a bacterial cell, little single-celled organism, and there you go. There's the little guy right there, and let's give him a little, there we go, and it says, hey, I need to make a copy of myself. I can actually turn into 1 billion copies in about 10 hours. For some bacteria, they can go through this process of duplicating themselves in about 20 minutes. Think about food poisoning. You went out for lunch, ate somewhere, within an hour or so, you're sick. Vomiting, diarrhea, stomach cramps, etc. That's because you ate a bunch of bad bacteria. Your body didn't like it. It doesn't work in your digestive system. The bacteria are replicating at an extremely rapid pace, and their population grows rapidly, or what we call exponentially. And this creates your health problems, your digestive issues due to food poisoning. Symptoms may mimic the flu, where you feel run down and just, I feel horrible. But think about the onset. The flu, you wear down over the course of several days before you get to that point because it's a viral infection. Food poisoning, bacterial infection. This is why we always worry about E. coli. And again, the key is how rapidly this infection can set in because of binary fission. So the general process of binary fission, you got your single-celled organism, your bacterial cell right here, and the chromosome attaches to the cell wall and doubles its information. The two loops of DNA get pushed apart. The cell wall begins to grow inward, cytokinesis. Okay, so here we are starting to grow inward from both directions, and it continues to pinch inward as the loops of DNA get pushed to opposite sides. So our bacterial DNA is over here and over here. Continue to push it apart and eventually pinch that cell in half or completely divide the cell. Bang! 2 cells, 20 minutes. Ideally, they should be duplicates, exact replicas. This is why it's similar to our cell cycle and the process of mitosis. So you got 2 cells. 20 minutes later, you're up to 4. 20 minutes later, you're up to 6. Keep doing the math. A doubling every 20 minutes makes a lot of bacterial cells. Okay, so they should be ideal duplicates. Now, bacteria do have other tricks, other methods of reproduction, of making copies or passing genetics. One other one to mention here is a thing called conjugation tubes. This is going to be what we will call bacterial sex. They're exchanging, or sometimes it's just passing a piece of DNA from one cell to another. Alright, so here's our example. We'll call this bacteria A, and this is bacteria B. Let's say these little hairy things, they're actually called pili, little hair-like structures that bacteria use to attach to stuff. Let's say this helps the bacteria survive because it can attach to whatever structure it's living on. Maybe that's part of our body or part of whatever. Well, that code to be able to produce that little structure is written in the DNA here. This loop of DNA, there's going to be a section that tells the bacterial cell how to make those little pili. So this little loop here, this little circle is known as a plasmid. That bacteria can take the information, put it into that circle, and pass it through one of these little conjugation tubes down to the bacterial cell down here. Now, the bacterial cell down there has a different coded DNA. Still got this big loop, but what's going to happen here is the yellow DNA, the plasmid, is given to bacterial B. That plasmid then becomes part of bacterial B's DNA code, and then bacterial B can now start producing its own little pili. Ooh, I need to attach to stuff. Now I can do it because I have the genetics from my friend bacteria A. So pili may not be a big deal. Who cares? They can attach. Whoopee! But consider antibacterial resistance or antibiotic resistance. If bacteria A has a resistance to penicillin and it passes it over to B, it's just double the number of individuals that are resistant. This is how resistant, one way resistance to antibiotics can spread through a bacterial population. Part of spreading is you have a resistant bacteria, it reproduces resistant bacteria and makes more resistant bacteria, but they can also give that resistance to the existing bacteria in the population. Okay? Alright, so the last thing, just a stupid trivia fact for you about genetics and cell cycle and making copies of ourselves. Hair. Hair is the second fastest growing body tissue. We have approximately 110 feet of hair growth every day. Right, and that's not just one big hair coming out of your nose or your ear. Think about, you shave your face, your legs, your wherever. How often do you have to shave? And you go, oh, I'm just cutting off all these little hairs. Well, yeah, little hairs, multiply that by thousands and tens of thousands of little hairs. How many feet of hair growth did you cut off every time you shave? Every time you cut your hair, go to the barber or the salon. So hair is going through that cell cycle. It's making an exact duplicate. Now, right now your hair might be red or brown or black or blonde or whatever. Later in life, it will start to change. Or, oh, I was born blonde, but now I have brown hair. Later in life, you're going to start to see those gray hairs showing up. They're changing color because the piece of DNA for color, the gene, is going to be red. So right now, let me give you a little visual here. Here's a strand of DNA. Right now, to give you black hair, this section of your DNA is being red. That's the gene for black hair. But later in life, as we get older, or stress, or environment, or whatever, this piece of DNA is going to be red. And now that's going to express the gray color in your hair. So it's still going through the same process of replicating. Everything's the same, but the gene expression shifts from the black section of the chromosome to the gray section of the chromosome. You inherited that information from mom and dad. Look at them as a good example of what type of hair color you're going to express later in life, when did they go gray, when should you think or possibly expect to start going gray. It's all written in your DNA. And hopefully as your cell goes through the cell cycle, everything is done correctly. Again, the key is checkpoints, proofreading the DNA, making sure the cell's doing everything right. Hopefully nothing is messed up. All right, so that's the cell cycle, cellular reproduction information. We're going to start moving into genetics and meiosis and how our body makes sperm and egg. So please keep in mind when we're looking at these cells here, all the cells going through this process are known as somatic cells. These are general body cells that are going through this process. All right, stay tuned. Let's check out meiosis.